great pleasure to open the session 15, Biosensitive Approaches, and to introduce my colleague, Alina Vasilescu, research professor in the International Center of Biodynamics, that would tell us some more on ways of coupling research with enzyme inhibition as a path for the selective detection of the fungicide thiram.
Thank you very much, Mihaela, and good morning. It's a pleasure to be here and be able to present my results after so many very interesting presentations. And my talk today deals with something that was the common denominator of many presentations here at the symposium, namely the search for selectivity, whether it was for imaging or for detection purposes. Here we focused on the enzyme inhibition and its coupling with the surface enhanced Raman spectroscopy. But although we were focused on an application relevant for the control of food quality, namely the detection of residual traces of fungicides in food products, this enzyme inhibitor system, it's relevant also for other applications because we know the importance of enzyme inhibitor systems in the biomedical field. The thiram is a dithiocarboamate fungicide which is no longer approved to be used in the European Union, and there are some strict limits imposed for imported fruits and vegetables. These limits are very tight compared with the maximum residual limits in other parts of the world. So, of course, we need good methods to detect such a small amount. Dithiocarboamate fungicides, from which thiram it's a part, are relevant not only in agriculture as fungicides, but they are also used in biomedicine. Some of them are already used as drugs or are studied to be drugs for anti-cancer or for other therapies. Among the properties of dithiocarboamate fungicides is also the fact that they inhibit some enzymes called aldehyde dehydrogenases, and based on their inhibitory action on these enzymes, there were drugs that were already used, such as antalcohol, which is based on the dithiocarboamate disulfiram and is used for the treatment of alcoholism. But also there were biosensors developed for the detection of dithiocarboamate fungicides based on the inhibition of the yeast aldehyde dehydrogenase. There are some challenges in the detection of dithiocarboamate fungicides for the selective detection, especially because they are degrading very fast. They have very much similarity between the compounds in the same group. So basically, the most official methods for the detection of dithiocarboamate fungicides are based on their acid degradation to carbon disulfide. So we can only detect them as a group and not individual. Or there are some chromatographic methods that can detect groups of dithiocarboamate fungicides, but not individual one. So for individual compounds detection, only very few methods are available. We set up to develop such a method for thiram, a selective method, and we wanted to use SIRS because, as Professor Ashtilan presented, it is a very sensitive and specific, provides a specific spectrum for a compound. However, there were few demonstrations so far where SIRS was used to determine an inhibitor by providing both its amount based on the inhibition and directly its amount based on its characteristic spectrum. As I mentioned, dithiocarboamates are among the inhibitors of aldehyde dehydrogenases, and there are some structures. This is disulfiram used in the treatment of alcoholism. Thiram, yaram, which are from the same group of dimethyl dithiocarbomates, then maneb and propineb. And so what is different? So here we have methyl, we have ethyl, here we have propyl. So these dithiocarbomates different in structure. The enzyme that we use to determine these compounds is a new enzyme that was isolated from an Antarctic bacterium. But as all aldehyde dehydrogenases, the reaction that it catalyzes is the conversion of aldehydes into carboxylic acid. And to do this, it needs a cofactor, which is called nicotinamide adenine dinucleotide. And in this catalytic process, the cofactor is reduced to NADH. Mainly is this NADH, which is used to measure the activity of this enzyme because it has a different spectroscopic, fluorimetric, and electrical properties compared to the oxidized cofactor. So this is just a little bit about this flavobacterium from which this enzyme was isolated. So it's from the Antarctic seawater, and it was isolated and expressed in Escherichia coli by our colleagues from the Institute of Biology. It was characterized in detail quite recently. It's an enzyme that has 55 kilodalton. It has a substrate binding domain, a cofactor binding domain, and a oligomerization domain. It was characterized, as I mentioned in details, regarding the kinetic parameters, and also some inhibitors and activators were screened. And this includes some heavy metals, which are active inhibitors. Regarding the enzyme inhibition by thiram, we set up to verify what is the mechanism by which thiram inhibits this enzyme. So we performed spectrometry and Raman studies, and we also did mass spectrometry studies. Following this test, we were able to determine that the mechanism of inhibition is a reversible one. And actually what is happening is that a mixed disulfide is formed between the thiram and the cysteine residue in the enzyme. So the enzyme is irreversibly inactivated, and this is very relevant from the point of view of developing an assay, because since the enzyme cannot be regenerated, our interest is to make the assay, because it will be single use, to make it as economical as possible. So use as little enzyme as possible with the best results. So this is basically what is the reaction mechanism. The enzyme binds the cofactor, then the substrates forming these complexes. Enzyme cofactor and substrate dissociate into enzyme product and reduce cofactor. But and this dissociate liberates the product, and the enzyme is reformed and able to re-enter the catalytic cycle. However, in the presence of an inhibitor, the complex enzyme inhibitor is formed, which then goes into inactive form. So the enzyme has no chance to regenerate. We measure the inhibition constant, which is 1.28 micromolar, meaning that thiram is a moderate inhibitor of this enzyme. So we can compare with other inhibitors for other enzymes. And just to see that this is also relevant for another field, not only for agro-food. The sulfiram was and some structural analogs were investigated for the selective inhibition of some enzymes in the human body for treating different diseases. So actually, these inhibitor studies of aldehyde dehydrogenases is very relevant for that field as well. We set up to make a very simple test for the detection of thiram and xyram by spectrophotometry, and it takes less than 25 minutes. And we applied it to measure the traces of thiram from the surface of spiked tomatoes. So we basically put thiram on the surface of tomato. We just use something to swipe the surface and analyze what we recover from there. And we used concentrations of thiram that were below the maximum limits in the European Union. Then, but of course, there was not necessarily a problem, but we know that from the start that the aldehyde dehydrogenases are inhibited by many compounds, right? So we know they are generally inhibited by the thiocarbamates, so by thiram, by thiram. We know that they were inhibited by heavy metals, right? So we can only apply this assay if we know for sure that in that sample we have thiram or thiram. But is not, if we don't know what we have in that sample, we cannot measure quantitatively what fungicide we have there. So then we said, okay, we are going to use cells because cells provide a characteristic spectrum. So what we used a very simple approach with silver colloids that were mixed with the sample and deposited on a filter paper. And then this filter paper was illuminated with the laser and we obtained the cells spectrum. This is the experimental setup. So this is the paper where we deposit the sample and this is the laser probe. Regarding thiocarbamates and their cells analysis, this was very much investigated and is well known that these thiocarbamates have high affinity for silver and gold surfaces. And what is happening is that this sulfide bond is cleaved and they bound, and the thiocarbamate ion that is found, that is resulting, it's binding to these surfaces in various configurations. So this makes that the cell spectrum is different than the Raman spectrum because of this. So we can follow the success of these modifications directly through the Raman measurements. These are some bands that are characteristic to disulfide and to other changes that occur when cleaved and absorbed on the surface. So, as I showed previously, the structures of other thiocarbamates, we can see that those who lacked the disulfide bond, like nanopropanol, have very different spectra compared with thiram and thiram, who have very, very similar spectra because they have also very similar structures. So, at the first view, it seems like it will be very hard to discriminate between thiram and thiram, even if we use cells, right? First, we did okay. We said, okay, let's first see how low we can go with this method because we use like very simple colloids and very simple method. So we can go up to nanomolar levels of thiram, and we can measure from again from the surface of spectrometers very well. It is very fast and it's more sensitive than the spectrophotometric assay. But if we just compare the spectra from thiram and thiram and using a simple visualization method like principal component analysis, we can see that the spectra from thiram and thiram overlap. So we need to, if we really want to make a difference between them, given the similarity between the spectra, we will need better classifier method. We need classifier method because this is not classifier method. So we have used partial least square discriminant analysis. And we had a set of 247 spectra, from which 198 were used for calibration and cross-validation, and 49 spectra were used just to test, to predict if we had thiram or thiram in them. And from these 49 spectra, 3 samples were misclassified. So the sensitivity and specificity of this model were quite good. It seems that if we are going to make a bigger database, then we can do this selective analysis of thiram and thiram by this method. But still, this means we need to do this data interpretation. So it's not simple, it's not straight away, it's not by simple PCA. Then, okay, since even by cells is not very simple, why don't we mix cells with enzymatic inhibition? So first, we analyze the spectra of NADH, the product of enzymatic reaction at different concentrations. These are the spectra of thiram at different concentrations, and these are the spectra of enzymatic mixture at the end of the enzymatic reaction in the presence of different amounts of thiram. So we can see how the bands are changing. This is for thiram, this is from NADH. So we can see the different ratio according to the different amounts of thiram present in the reaction medium. Basically, the same thing can be made in the case of thiram. So if we analyze the ratio between the intensity of some of these bands, we can correlate it with the residual activity of the enzyme. So we have a search-based method to use instead of a spectrometric one to evaluate the inhibition of aldehyde dehydrogenase. When we analyze all the spectra of enzymatic mixture that contain thiram and thiram, but principal component analysis, we see the same thing as when we have individual samples of thiram and thiram without enzyme, an overlap. But if we, so this is just looking at the bands for thiram and thiram, fungicide-specific bands. But if in this analysis we also include three bands that correspond to NADH, then we improve this separation, even with simple visualization by PCA. So basically, this is as far as we went with this approach so far. In conclusion, the irreversible inhibition of NADH by thiram can be used for practical purposes to determine the fungicides from the surface of tomatoes and other vegetables. There is a potential to integrate in some low-cost devices to be used for the analysis of agro-food products that are appropriate for cold storage, because this enzyme is a cyclophilic one and it has activity even at low temperature, but this requires further development. We monitor the enzymatic activity by cells instead of spectrometry. We confirm that by combining cells with enzymatic inhibition, we can increase the selectivity. And of course, all these analyses that were presented were on solutions of thiram and thiram analyzed individually. We didn't have any experiments yet with mixtures of thiram and thiram, and so this should be a next experiment. But we need to also, we can do other improvements in these systems. For example, we can integrate some mediators for NADH, which have very strong Raman signals to boost the sensitivity. And beyond the detection of thiram, we can apply this procedure for other systems of enzyme inhibitors, which are relevant, as I mentioned, not only for agro-food, but also for the biomedical field. I would like to acknowledge the funding, and also I would like to acknowledge the advice from Professors Didier Fournier and Jean-Louis Marty regarding the enzymatic inhibition mechanism, and all these collaborators which were co-authors to our paper, and I didn't include here their results, but they were related to modeling and docking studies, to chromatographic separation and detection of recovery from spike tomatoes, and to the characterization of the colloids that were used in the cells measurements. So I just want to emphasize that in addition to the many groups from Romania that were already on the first slide, so it was a collaboration with USA, South Africa, and Spain. So I think this is very much in the spirit of this symposium, and shows that when we collaborate and put expertise from many fields together, we can obtain much better results. So I'd like to thank you very much for your attention.
Thank you very much. And if questions appear from the audience, I think Alina will gladly take some.
I have a question regarding the data. So you said that when you included some other bands from the spectra, you increase the separation between those two groups. So if you will include the entire spectrum, maybe this would increase the sensitivity?
No, we did the verification. So if we just ignore the bands from NADH and include the whole spectrum, but just without these three bands, the separation is no better than just the bands from time. So it's absolutely critical that you include these bands to have the separation.
Only those bands, yeah. Okay.
I obviously don't know in which category these substances go, but are they part of an interest for forensic science, like they are illicit compounds which are not allowed to cross borders. These are of interest to forensic science.
In principle, CERS is very
I know,
used.
but I refer to the you sensitize the method for this compound. I'm wondering if this could be labeled as of interest for forensic science. It's not.
I'm not sure. I didn't read so much about forensic science, so I'm not very familiar, except that CERS is very much used in this field.
Yeah, I'll be interested to find out.
One more question?
Yeah. Ready? No, please. Well, for me, thank you very much. I'm ashamed that it's the first time that I attend this presentation, Alina. Question. If we have these overlaps that you pinpointed, is it so wrong? Because I think that the most important thing is to detect that in on a tomato, whatever the sample, there is something that it shouldn't be there.
It is, no, the limits are different because the toxicity of the compounds is different. So the limits are different depending on some compounds, but there are also some limits for total carbon, just because it's very difficult to realize.
My comment relates to the following idea. It's a detection that means a flag should be raised. Well, there is something that it shouldn't be, and then to start to detail. So the idea is, instead of trying to detect all the details, just to flag that there is a class of compounds that may, well, of course, if the case should be then precisely, specifically analyzed, but not necessarily to start deploying an army of more or less rather complex methods. So that was, because CERS seems to be, well, we have, thanks to you. So I was thinking about a multi-tiered approach.
Well, it's very easy just to make an enzymatic inhibition like this. It's just if you want to have just everything. Or CERS also, you don't care if you don't want to know what you have there. You just make a spectrum and you say, okay, it's a thiocarbamate or something. But because their toxicity is different, you would like to use all the powers of CERS. I mean, it would be a waste not to use it, in my opinion.
Yeah, but I was not thinking about the analytical perspective, but the layman. So if I take a tomato, I would like to know that it's okay for me to take it. Yeah, okay, thank you.
Ready?
No, I was just looking, right?
Okay, you have the back problem like yesterday. Thank you very much, Alina.
Thank you.